Hello there. Good morning, good afternoon, good evening, depending on the part of the world you are listening to me from. Welcome to another lesson in the BET series, Biologists Explain This. I still remain your biology teacher, Peter Sapiodun, and today we continue our discussion on enzymes. This is the third in the series of our discussions on enzymes. Our first lesson had to do with enzyme activity. We talked about structure of an enzymes, how enzymes are specific. We talked about energy of activation. We talked about the concept of the enzyme being denatured. And the second lesson, we talked about the models that explain how enzyme and the substrates they fit together. We discussed the lock and key model proposed by M. A. Fisher in 1894, and then we talked about the model proposed by Koshland, in 1958. So we are discussing today the issue of effects of substrate and enzyme concentration. Of course, we've said previously that there are a couple of factors that might affect an enzyme's efficiency or effectiveness in catalyzing a reaction. So today's lesson is focused on those factors. What are the factors that will affect enzymes activity? And we are discussing two of them in today's lesson. So come along and let's enjoy this lesson together. Now let's look at our learning objective first before we proceed. By the end of this lesson, you will be able to describe the effect of substrate concentration on enzyme activity and be able to describe the effect of enzyme concentration on enzyme activity also, and be able to explain what Vmax and Km are and their relevance to enzyme activity, and be able to explain Michaelis-Menten kinetics, and then lastly, of course, be able to appropriately apply this concept you will have learned to relevant and requisite situations as the occasion may demand. So, having seen our learning objectives, we turn our attention to the issue of this lesson for this particular presentation. Now, let's look at first off substrate concentration. Of course, there are four factors that can affect enzyme activity. You've got substrate concentration, you've got enzyme concentration, and then you've got temperature and pH respectively. So we are focusing on the two concentrations in this lesson. Now, when you increase substrate concentration, of course, one thing I want to let you know from this lesson is to always remember that since we are talking about four different variables that can affect an enzyme's activity, when you are discussing one of these variables, others have to be kept constant, or let's say assumed to be constant. It's very important. You can't be having to vary two of these at the same time. To control pH and temperature at the same time with the same efficiency will be very difficult. Of course it's possible, but it's very difficult. So usually, if in science, if you're doing any study, you keep all other variables that can interfere with your result, you keep them constant. So if we are focusing on substrate concentration, we have to ensure or endeavor that enzyme concentration, temperature, and pH are constant, or as they are favorable or as optimum as possible, not to interfere with what we are trying to find out. Our focus is just on substrate alone. So that will apply to other variables too. So we get the temperature. We are supposed to keep pH and the two concentrations respectively constant or optimum. So now when temperature increases, definitely the kinetic energy, that's the energy of motion, is increased. Both the substrate and enzyme molecules acquire increased kinetic energy when temperature increases, and of course when they are energized, they are able to move more around, they become more active, and then they collide more often, and these collisions, they are likely to lead to product formation. Of course, not all of these collisions will be effective, but most of them will be effective. As temperature increases, virtually all the molecules in the reaction will have acquired enough kinetic energy to be active enough to combine with the others, to form the product. So it's important to understand that there is this this pathway that we have when an enzyme is reacting with a substrate, there's this transition state called the ES complex, the enzyme substrate complex. That if ES complex is not formed in a reaction, it shows that no reaction is going on. That shows an affinity between the substrate and the active site of its enzyme. So always remember that the ES complex is very important. For that is the target of some inhibitors, as we are going to see in subsequent lessons. So when you have increased kinetic energy of the molecules, you form the ES complexes more quickly, meaning faster, and then of course rate of reaction increases. Okay, so we are looking at the issue of the effect of temperature, and it's very important to understand that there are many more free molecules. Okay, as you have the molecules being active at higher concentrations of the substrate, the enzyme molecules become more saturated. Now we are not discussing temperature today. Okay, I'm just trying to prepare your mind for what's ahead. Now, in discussing the effect of substrate concentration, as you have increasing substrate concentration, more and more substrate molecule. Remember, the enzyme is fixed. Very important in this situation. If the enzyme is fixed and you're increasing the substrate, of course, more and more substrate will occupy the available fixed enzyme active site. Okay, and then it's going to get to, so at that stage the reaction rate will increase. So in this stage now, the rate of reaction is directly proportional to the concentration of the substrate, of course, but it gets to a point where the active sites begin to get crowded, and at a point the active sites become saturated with substrate molecules, meaning no further substrate molecule may be able to fit into any of the active sites available. Remember again, the enzyme is fixed in its concentration. So just imagine that if you are having more, let's say you have a family of just one room, okay, or two bedded, self-contained, and things like that. So you have the dad and the mom, and then you have one child, and suddenly the second child came, and then you're having three children and stay in those two rooms. There's going to be a problem eventually, because you're having, the family is growing, but the space available remains the same. The same thing if you have an organization or a school, for example, a school that rented an apartment to run the school, and suddenly you're having 10 students, you have 10, 20, 30, and then just in that space, it's going to be, each classroom becomes crowded, and the students can never enjoy it because it's going to become unconducive, very inconvenient for them. Toilet facilities become a problem and things like that. So you will need to look for another space or to build your own permanent site, as it is called. So use that analogy to understand the concept of the enzyme being fixed or the substrate increasing. The rate of reaction will not increase indefinitely. Initially, as long as there are still more empty active sites, the rate of reaction will increase, because with increasing substrate, there will be more collisions among the substrate molecules, which will lead to product formation. But as we begin to have crowdedness, it becomes an issue. The molecules cannot move around anymore. There are fewer collisions, fewer ES complexes being formed, and hence the reaction will stagnate at a point. Remember, it's a stagnation, meaning reaction is still going on, but that's the best the enzyme can do, because there are no other spaces, until when the present substrates have been all converted to product, and all the products can be released from the active site. The active site is now free for more substrate molecules to fit into all the active sites that are available. But until you have converted all the substrate to products, the reaction rate remains constant throughout. And look at the next thing. So we look at the next thing. Look at this graph. The graph is clear enough. Just take time out to study. You see that the graph increases and then it plateaus, meaning it gets the peak at the point, because at that point when the graph becomes parallel to the x-axis, all the active sites have been occupied as at present, and so until all the substrate have become products and released from the active site, the reaction rate remains the same. And so let's look at still on the effect of concentrations. We turn our attention now to enzyme concentration. Now look at enzyme concentration. Again, if you're studying enzyme or the effect of enzyme concentration, we have to keep substrate constant, meaning substrate concentration has to be kept constant. So how do we do this now? Now you can ask, how do we do this? Now, to prepare enzymes and substrate, you have to weigh a particular mass. So you weigh a particular amount of the substrate on the balance, and then mix with a particular amount of water to give you the concentration, maybe 2 molar or 10 molar or 5 molar. And then the same thing you do for the enzyme. You weigh the enzyme and dilute in a particular amount of water to get you the concentration. So those are standardized, as you know. Okay, so you know the mass that you dissolve in a particular volume of water to give you a particular concentration. So you make the substrate constant when you are focusing on the effect of varying enzyme concentration, and of course the temperature and pH also will remain constant. So when we are focusing on enzyme variable now, okay, we have to keep all the other 3 factors constant or optimum as possible. So if you look at it, let's look at the analogy here now as we did in the last one. Let's use the example of a school. So you are the proprietor, for example. You rented an apartment, and then you've got these few students in a class, maybe not more than 5. And then maybe you just had the money, and then you just felt like building a new site, a large complex with so many classrooms, okay, even up to story building, top floor, and things like that. And then you moved the students that you had in this present building. They are not even enough yet. Let's say you have enough 20 students, and then you move these 20 students to the new site. And so let's say in a class you don't have more than 5. And the question is this: is going to be a problem, because when they even talk, there will be vibration echoes, of course, and then there are going to be large space to play around, few friends and things like that to play with. And so that's the scenario here. When you move, just like a bachelor, also a bachelor that's living in a one-room self-contained, okay, what you call a suite, is living in a suite apartment, and then suddenly he's not yet married, he's not got any siblings staying with him or her, and then this person moved into a duplex. Imagine one person in a duplex. What's the sense in it? What's the benefit of that? Not that you are married, or you are, even if you are going to marry. I mean, imagine a duplex of 5 rooms and you are the only one. Maybe you have a maid that comes and goes and things like that. Just imagine. So when the space, so the active side is the space now, that's moving from a 1-room and suite into a duplex of 5 rooms. When the substrates, which are students now, or that bachelor, the students remain the same, meaning the substrate concentration is fixed, but the enzyme is increasing. Now, usually you have seen students who came to me and talked to me about the graph they came across, that the rate against the enzyme is a straight line. Now let me say this with emphasis and clarity. Unless the substrate molecule was in excess before, okay, imagine that school I talked about, for example, the example of the school. Let's say your students are now growing and they have outgrown the spaces available. It's reasonable and ideal or favorable to move them into a larger space. That is reasonable because you now have more students than the space available. So remember that picture, that analogy. When the substrate is fixed and the enzyme increases, you are increasing more active sites. The question is what are you going to put in the active site? Without enough substrates, there will be not enough collisions, because when the enzyme increases in concentration, the structural molecules can fit into anywhere. Remember, it's collisions that lead to product formation. So if you've got few molecules in that, no active site is fully filled, and then the molecules of the substrate are just scattered, it's going to slow down the reaction. So note, initially, when enzyme increases in concentration and the graph is linear, it suggests or indicates that the substrate was in excess before. The moment the substrates have all fitted into the active site presently, any further increase in the enzyme concentration will result in no further increase in the rate of reaction. So please note that the graph of rate against enzyme concentration, it's not an indefinite straight line. It plateaus, meaning it gets to the peak at a point and levels up, just like the one that is typical of the substrate concentration also. And so we look at the graph illustrating the effect of enzyme concentration. You can see, like I explained, the 2 graphs are explanatory enough. Just go through them and you can relate them to the explanations that we gave to the other side before now. And let's look at the Michaelis-Menten kinetics now. Okay, of course, kinetics talk about progress, dynamism, and things like that. But some of these kinetic means there's an action involved. So these two scientists, Michaelis and Menten, they are two individuals. Their researches combined together give us this concept. And then the explanation was that was to give us a picture of how the rate of an enzyme controlled or an enzyme catalyzed reaction depends on the concentration of both the enzyme and the substrate. Okay, now if you consider a typical reaction like we said previously, you form an ES complex when a particular substrate is complementary to the enzyme and the substrate fitted into the active site of this enzyme. The ES complex, which is called the transition state, was formed, and then you go on to product formation as you see below the screen there. So you go for E to A plus S to ES, which is ES complex. Then from there you even form the EP complex, meaning the ES and the EP are both transition states on the way to forming the product. Of course, when the products are formed, they are not compatible or complementary with the active site. They got to be released, and the active site is free. That's why we always say that enzymes can be reused, because they are not consumed or destroyed. Once the products are released, the active sites of the enzymes are free again for substrate molecules to bind with them. Now, still on Michaelis-Menten kinetics, there are two important variables or two factors that we discuss under these kinetics. Now, first is the concept of the Vmax. Now, the Vmax, of course, you're familiar with V from your physics studies under motion. V talks about speed. Okay, if it's a scalar, it talks about velocity, if it's a vector, as it were. Now, Vmax is talking about the maximum rate of a reaction when all the enzymes active sites are saturated with substrate. Remember our previous explanation, okay, in previous slides. As the substrate is increasing, there are more collisions of the substrate with themselves and with the R groups in the active site, and ES complexes are being formed, and the products are being formed as a result. So you get to a point where the enzyme is working at the maximum possible rate, that point where the graph plateaus and then levels off and becomes parallel to the x-axis. So that maximum is what we refer to as the Vmax. It indicates that all the active sites have become saturated with substrate. No more space for any more substrate molecules. And the Km, called Michaelis-Menten constant, what it indicates is this, that this constant talks about when an enzyme is working on the substrate, meaning there is dynamic progress going on. When there's a dynamism, you get to a point where you can determine the rate of reaction at the early stage of the reaction. Now, it's always best to determine rate of reaction within the first few seconds of a reaction. So enzyme reactions are fastest at the early stage of the reaction. So what we are discussing is this now, that the Km is a concentration. Now, whose concentration? Km is defined or described as the substrate concentration that produces 50% of the Vmax, that is half of the Vmax. Again, let's define it. The Km, Michaelis-Menten constant, is the substrate concentration which gives half, that is 50% of the maximum rate, which is the Vmax. Now, the Km is a measure of the affinity. So the importance of the Km is that it gives us an indication, it sheds light on the affinity between the substrate and its enzyme. Is the affinity strong or is it weak? So what we are saying is that when you have a low Km of an enzyme's activity, it shows that the enzyme binds well, meaning the enzyme has a higher affinity. So what we are seeing here is mathematically affinity of the enzyme for the substrate is inversely proportional to the value of the Km of the enzyme. Let me take that again. The affinity of the enzyme for the substrate is inversely proportional to the Km value of that enzyme. So in other words, as the Km reduces or decreases, the enzyme binds well. So a good enzyme, okay, has a low Km. That is the picture that we're painting here. And then let's come to Vmax. Let's pick this concept individually as just a few more details. So, like I said earlier on, Vmax is the rate of reaction when the enzyme is saturated with substrate molecules, of course, and then it shows the highest turnover, meaning the maximum rate of product formation. That's what you call turnover. How fast are products being formed or produced in the catalytic process? And of course, that relationship is is premised on the fact that the substrate molecules are binding very well with the active site. So if you look at that, the relationship between the rate of reaction and the substrate concentration is dependent a lot on the affinity of the enzyme for the substrate. And something more about Km, you said Km is the substrate concentration that gives half, that is 50% of the Vmax. Okay, it's always in the case. So just for the sake of emphasis, never get tired of listening. That's how children learn. A for apple, B for boy. Okay, I mean a child can repeat and repeat the alphabets, can repeat the letters without even knowing them. Just keep them because repetition enhances memory and understanding and recollection, of course. So you're just saying this for the sake of emphasis that Km is an indication of an enzyme's affinity for its substrate. The lower the Km, the higher the affinity of the enzyme for the substrate. And still on Michaelis-Menten constant, just look at that graph, study the graph. You can see the peak and you can see how Vmax. So you can, if you are asked in an examination, how can you obtain Km from a graph? It's so simple. Let's say the peak Vmax is 10, for example. Now on the y-axis, take 5. 5 is the half of 10. Now extrapolate your 5 from the y-axis and trace it to join the curve. And then trace it down to the x-axis where you have the substrate concentration. Let me say that again, graphically, because there's a mathematical formula for obtaining or calculating Km. But from the graph, graphically, you can get your Km when you know the Vmax. Hypothetically, I said the Vmax is 10, half of Vmax is 5. Connect your 5 to the graph, trace it down to the x-axis. Where your line intersects with the x-axis, that is the Km. And how do you determine Km and Vmax? Okay, if you are going the route of mathematics, okay, now, or let's say, okay, of course, you have to carry out a procedure in the laboratory, and then your data will be plotted as a graph. Now, if you incubated different enzyme concentrations, okay, mixed with enzymes, now what you are going to get is that when you determine when you get your value, you plot it on the graph. Okay, you plot rates of the reaction on the y-axis against the substrate concentration on the x-axis. What you're going to get is that the graph is that we saw is hyperbolic, is linear initially, then levels off. That's the hyperbolic nature of the graph. But you can subject your results to mathematical analysis. So your V, okay, the rate of reaction at every point of the reaction. So if you are looking at determining your rate at every point of the reaction, every step, your rate of reaction at every instant of the reaction is given by Vmax divided by 1 plus Km divided by substrate in brackets. So you can imagine the mathematics there. So that's why a more convenient way to obtain your Km is graphically. And then, of course, we say that Michaelis-Menten plot or graph is hyperbolic, okay, not a straight line. Now, it's always difficult when you don't have a straight line graph to explain a theory or a concept. So there are 3 other models, okay, let's say 3 modifications of Michaelis-Menten plots, but we just talk about one of them, which is the most acceptable. It's called the Lineweaver-Buck double reciprocal plots. Okay, it rearranges. What it's talked about is that there's an inverse of all the variables you have. So instead of v, you've got 1 over v is equal to, instead of v max, you have 1 over v max, and like that. So it's an inverse plot. So if you look at it, when you plot 1v, that is the inverse of the rate against the inverse of the substrate, what you're going to get are the parameters you see on the screen there. On the y-axis, you plot your inverse of the rate, and then when you plot the graph, you're going to get the gradient and then the intercept. And then let's look at, now if you look at what you are going to get, so if you plotted that on the graph, now this particular graph tells you what, so that line over buck, okay, reciprocal plot gives us this graph. This is a more linear graph. Of course, it has its own disadvantage. It's just simple, okay, it makes explanation easier, but there's more weight on the individual values, because that's a great disadvantage. Meaning you have to keep looking at how to have a perfect figure, perfect data. So that's a lot of burden. That's a great disadvantage of this kind of plot, but it's simple enough. So when you plot your graph, you get your Km is going to be obtained as an inverse. So where the graph intercepts the x-axis, that is your Km, so the inverse of the Km. So you can obtain your Km graphically from the Vmax, half of that, or you can use the Lagrange double reciprocal to obtain it mathematically. So please note that. But also you can also obtain your Km from the graph, from the Lagrange double reciprocal graph. But like I said, it's always more convenient, but it depends on the kind of study you are embarking on and determine whether you are going to use the Lagrange modification or the Michaelis-Menten model, just the way it is, like that. So we are looking at the Michaelis-Menten constant again. Okay, like we said, the equation describes the initial rate of reaction. Okay, that is V naught, and it's affected by the substrate at that time. Now, I said it previously that it's very favorable and much more ideal to determine an enzyme rate of reaction at the early stage of the reaction, okay? Because enzyme reaction are fastest at the beginning, because at that time, rate of reaction depends entirely on the available active sites. The moment the active sites become crowded gradually by substrate molecules, it's no longer the substrate molecule that is the limiting factor. Remember the concept of limiting factor. If a process depends on more than one variable being relevant or impactful, then the rate of that process is determined or limited by the factor of variable that is nearest its lowest value. Let me say that again. Okay, if you want to define limiting factor, okay, it talks about the fact that if a process depends on more than one variable being relevant or being impactful, the rate of that particular process is limited by the variable or factor that is nearest its lowest value. Let's say, for example, you want to travel, you have planned everything, okay? You bought fuel, you did everything, checked your engine and everything, and just in the course of the journey, one of your tires deflated or burst. You can imagine every other variable, engine oil, radiator water, everything was okay, but just one thing, out of 4 tires, one tire bursts. That tire singularly will slow you down for the next minute or hour, because if you've got no vulcanizer nearby, you're going to be stuck for hours. One tire has limited your progress. That's the picture that you have there. So when you look at the whole thing at the beginning, so please note, enzyme rate of reaction is best determined at the early stage of the reaction, because that is when the reaction depends basically on the available active sites, meaning the substrate is the limiting factor. Then remember, when you are discussing substrate concentration, you keep the enzyme constant. So there are still available active sites for the substrate to fit into. So there's enough space. Remember, enzymes are bigger than the substrate molecules they catalyze. Let me repeat that. Enzymes are bigger than the substrate molecules decatalyze. One single enzyme can bind to hundreds of thousands, even millions of substrate molecules at the same time. So please note that fact. And so in the discussion of Michaelis-Menten constant equation or kinetics, you've got the issue of when the part of the graph was straight on the plot, you call that the first order kinetics because it depends on the substrate concentration, and of course it tells you how the whole process depends on an increasing substrate concentration. By the time the graph levels up, becomes parallel to the x-axis, you call it the zero order, meaning no progress is being made. There's this constancy. So when the graph plateaus, gets to the peak, you have an increasing substrate, has no effect whatsoever on the rate of reaction. That's why we call it the zero order kinetics. No progress is being made. Same thing the enzyme is doing is what it's doing for the next couple of seconds. The enzyme is working at the same rate. Nothing can be done whatsoever at this stage to increase the rate of the reaction. So let's look at the importance of determining Michaelis-Menten constant and the Vmax. Now, the relative concentration of the substrates under normal conditions, it helps us to be able to predict whether or not the rate of product formation is actually affected by the available substrate concentration. Since you're plotting a graph, you can look at the graph because a graph tells you how two variables are directly influencing each other. So pick, for example, if two enzymes in different pathways are competing for, meaning they depend on that same substrate. Of course, the question is, which of these two pathways will progress or proceed? Just imagine these two pathways need these substrates as a necessity. Okay, it is the value of the Km and the Vmax respectively that will let us know which of these pathways will be more feasible, meaning which of them will use the substrate that's available much more efficiently and much more easily. That's important. So in practical terms or situations, it's always good to know the value of Km and Vmax so that you can know which pathway, because there are hundreds of reactions going on in the cell at the same time. Like I said, the cell is a closed system where all reactions form a pathway being dependent on one another. So look at the graph, study the graph that you see to know what happens when you have a low Km and a high Km. Like I said, low Km, value of Km is inversely proportional to the enzyme's affinity for its substrate. So a low Km means high affinity of the enzyme for the substrate. So as we round off, let's look at the assessment questions that you have. Okay, study them. You can go over the slides over and over again so that you can have a clear understanding before you attempt the questions. Still on assessment questions, study this question again. This is an extension of your knowledge. Sometimes in higher biology, in particular, if you are a Cambridge advanced level student, you won't expect straightforward questions. If you look at Bloom's taxonomy of learning, the highest height is what you call thinking outside the box, being able to create answers, being able to think of novel situations. So if you are given something that is not from the textbook, how do you apply it? That's the kind of question you are seeing on this page. Still on assessment questions, you see also this is also an application question. It's adapted from a past question in Cambridge Advanced Level Biology paper. And then of course you've got this a graph to help you to still apply your knowledge in application situations, not straightforward. So you've got to think very well, apply your concepts to this graph interpretation. You can see that there are a lot of questions. These questions are particularly focused on advanced level students who are having issues with graphs. Over the years, I've got a lot of my students complaining about enzymes. In the Cambridge Advanced Levels syllabus, the topic enzyme is very short, very small topic. but then it has application to virtually all aspects of biology. You see it in DNA polymerase, in replication of DNA, you see it in even cell division, mitosis, you see it in reproduction. It's everywhere. So it's relevant. That's why you seem to see more questions here based on application of the knowledge. So finally, sending your answers to my WhatsApp, to my email, to my Instagram and YouTube. I will be eager to attend to your answers and get back to you if there are things that I need to explain. And if you have questions, please don't delay. Don't hold back. We are friends now, okay? And then it's important that you tag your friends, your colleagues, your pals, okay? If you have enjoyed our lessons, you have been blessed with more understanding. Now you are clear, you are calm, the anxiety is gone. Let others also enjoy these lessons and benefit from them so that you can pass together that examination. So our goal in BET series is to ensure that you excel at one sitting, never having to resist an examination. So we wish you the best in your exams. Thank you for listening. See you next time. Bye.